Thank you everyone for joining us today for the digital launch of Bio-Rad's Pioneer Antibody Discovery Platform. The Pioneer platform has been designed for the generation of high-affinity, high-developability leads, and our speakers will explain how we've generated one of the most advanced human antibody libraries on the market today, based on Bio-Rad's decades of antibody experience, and they'll also cover how the antibody discovery process works. Before I introduce you to our first speaker, Dr. Francisco Ylera, I'd like to cover a couple of housekeeping items. You can submit questions for the speakers using the Q&A widget. These will be answered after the presentations. If you encounter any technical issues, you can click on the Help icon for a list of solutions to common problems. You can resize the webinar slides by clicking on the icons above them. Our first speaker today is Dr. Francisco Ylera, the head of the R&D team that generated the Pioneer Antibody Discovery Platform. Francisco has been with Bio-Rad for over 18 years and has a PhD in biochemistry from the Free University Berlin in Germany and postdoctoral experience at the Harvard Institute of Medicine in Boston, USA. He's worked on customer antibody generation projects to design the selection, antibody characterization, and technical support, as well as method optimizations. And he's now going to introduce Bio-Rad's Pioneer Antibody Discovery Platform. Over to you, Francisco.
Thank you, Kim, for the introduction. In this presentation, I will describe the Pioneer library design, explain why we call it a platform and not a library, and show data on its performance. I would like to start with a few words on the site where the platform was developed. This group was founded more than 19 years ago. Since then, we are generating custom antibodies by phage display as a service for our customers. We have a long history and a deep understanding of antibody libraries and selection systems, and now use this experience to generate a new library, the Pioneer library, which delivers therapeutic antibodies. We are excited that we recently moved to a new site, which gives us more space to expand for the new services. Pioneer is a highly optimized synthetic library. This has many advantages over other library approaches. If you look at a general phage display library, there is always a significant part of the library which does not deliver any antibodies after selection, shown here in light gray. This could be because of low expression of these sequences in E. coli, poor folding, or low display rate. This is especially the case for donor libraries. Thus, the library size given is usually far larger than the actual functional size of a library from which you can get antibodies. For Pioneer, we focused on maximizing the functional library space by using sequences which are well suited for phage display. Furthermore, codon usage is optimized for E. coli to get a high and well-balanced expression of all antibody clones. The advantage of a synthetic library is a controlled and basically unlimited diversity in the CDRs. Our heavy chain CDR3 design has a theoretical diversity of more than 10 to the 25 different sequences. Even though we cannot synthesize these all, it exceeds the actual library size massively, which means that it is extremely unlikely that two transformants contain the same CDR3. This makes the generation of large libraries simple and facilitates the pretty accurate estimation of its size. And here we looked only at the CDR3 of the heavy chain. For Pioneer, all 6 CDRs are diversified with a germline-specific design which is close to nature. The Pioneer library has a functional library size of 225 billion unique antibodies, which makes it the largest functional antibody phage display library ever made. It is significantly larger than libraries of our competitors. Library size is important, as the larger the library, the more specific antibodies you will get, with a better epitope coverage, higher affinities, and more antibodies to choose from for your lead candidates at the end. I will prove that with data later in this presentation. Another important aspect of an antibody library for therapeutic applications is the developability. For Pioneer, we use defined germline genes with favorable properties which are frequently found among therapeutic antibodies on the market or in the clinic. We selected 2 heavy chain and 2 light chain germline genes, which in combination make up the 4 Pioneer sublibraries which are kept separate. In contrast to natural antibodies, we do not have mutations outside the CDR regions, which are known to increase the immunogenicity risk. We also minimized amino acid motifs which could cause CMC concerns. For Pioneer, it is shown here in green compared to the natural antibody repertoire in gray. For the analysis of the natural antibody repertoire, we analyzed 700,000 heavy and 100,000 light chains of the same germline genes as Pioneer. You can see that in average, Pioneer antibodies have less motifs of concerns than natural antibodies. For the Pioneer library, we developed a new phage display selection system, SpyDisplay. It is based on the SpyTag technology, which consists of a small peptide tag, the SpyTag, and a small protein, the SpyCatcher, which, when mixed together, spontaneously autocatalytically form an isopeptide bond. This is a stable covalent bond and it is irreversible. The reaction is very fast and quantitative. Already after a few seconds, you can detect the protein ligation product. The reaction is specific for these 2 partners and does not happen with any other proteins. Therefore, you do not need to purify proteins, but it can also take place within cells. That is what we use in SpyDisplay. In SpyDisplay, we use a phage MIT which encodes only the Fab and no other phage display elements. We use a strain with a modified genome into which we inserted a SpyCatcher P3 fusion. After infection with helper phage to add the other phage proteins, bacteria generates new phages which carry a SpyCatcher on the surface of the phage. From the phage MIT, we have the Fab expressed with a SpyTag at the end. And then, within the cells, SpyTag and SpyCatcher ligate, and the Fab is covalently displayed on the surface of the phage. The phage MIT is packaged inside the phage, which leads to the linkage of genotype and phenotype. This method has several advantages. First, there is no subcloning step required between selection and expression, not even for large-scale expressions, as the library is already in the expression vector. This is faster, less steps means less error-prone. We even further accelerated the protocol to run 1 panning round per day. The Fab is expressed without the large P3 fusion. Since smaller proteins, especially mammalian proteins, fold much better in E. coli, SpyDisplay facilitates the display of more properly folded Fabs on the surface of the phage. Obviously, the resulting Fabs carry a SpyTag and therefore are compatible with our modular antibody assembly platform Trailblazer, which I will explain on the next slide. We showed that we have a truly monovalent display, that means not more than 1 Fab per phage, for the selection of high-affinity antibodies. But we can also switch to a polyvalent display to make use of avidity for the selection against difficult targets. The system has further advantages which are listed in our paper, which was recently published. In our modular antibody assembly technology, Trailblazer, we use our Fabs with a SpyTag at the C-terminus of the heavy chain in combination with a suite of SpyCatchers. We have monovalent SpyCatchers, dimers, the so-called BiCatchers, and SpyCatchers fused to Fc domains, the Fc catchers. Furthermore, we have inserted cysteines into these catchers for site-specific labeling. After a single Fab production, you can get access to a large range of different antibody formats. The coupling reaction has the simplest protocol you can think of. Just mix the antibody and the catcher, let it sit on the bench for an hour, done. No purification required. The reaction is fully scalable and can be done just for an assay or at larger scale. Leftover material can be put back into the freezer. Since the reaction is site-specific, there is no modification of the antibody binding site. Therefore, these coupled products usually perform better than conventionally labeled antibodies. The coupling reaction is so simple and fast, you can use the best label and format for each and every assay and quickly switch depending on your needs. Especially interesting for therapeutic projects is the synthetic IgG format. The whole selection output can be converted into this IgG format within 1 hour for IgG prototyping and an accelerated characterization. This is much more economic than the slow process of recloning into a mammalian vector and transient mammalian expression. Only the best candidates are then later converted into the real IgG and expressed in mammalian cells. To summarize the design part, the Pioneer Antibody Discovery Platform delivers a large and diverse set of hits after selection due to its large size and its high quality built in during construction, and because the library was optimized for phage display. The resulting antibodies have a good developability with a minimum number of sequence motifs which could cause developability concerns, favorable germlines, and a curated germline-specific CDR design. The outcome is a good proportion of antibodies with sub-nanomolar affinities, also due to our novel display system, SpyDisplay, which gives an improved performance, offers a faster selection protocol, and delivers antibodies which are compatible with our Trailblazer platform for accelerated antibody characterization. Now let's look at some data. We performed test selections against 4 targets, a mix of smaller and larger proteins. In average, 69% of the screened antibodies after selection were ELISA positive, with a signal of at least 10-fold above background. After sequencing portion of these hits, 58% were unique antibodies. The numbers for the 4 Pioneer sublibraries are shown in this diagram in shades of green. So we have a very good diversity after panning. Next, we looked at the heavy chain CDR3 length distribution because this CDR usually determines the bound epitope. We see a distribution over all CDR3 lengths which make up the library, which roughly follows the library composition. We also looked at these peaks, which are at different lengths for each antigen and therefore are not library but antigen dependent. Also within this peak, we observed a good diversity between the different candidates and not just an exchange of a single amino acid at an irrelevant position. We next determined the monovalent affinities by BLI. Basically, antibodies with affinities above 100 nanomolars are useless as they are unlikely to work in the characterization assays and therefore will not be identified as potentially useful. Antibodies in the double-digit nanomolar range might work in assays but would require affinity maturation to become lead candidates. The majority of Pioneer antibodies have affinities in the single-digit nanomolar range, which is sufficient for characterizing these antibodies and might even be sufficient for therapeutic antibodies. Of course, affinity maturation is always possible, but might not be required anymore. For all targets, we found a good number of candidates in the sub-nanomolar range. These affinities are in the range of therapeutic antibodies, and affinity maturation is usually not required. We even have some antibodies with affinities below 100 picomolar, directly out of the library. These are affinities which are better than those of most therapeutic antibodies. In average, more than 20% of the antibodies had affinities in the sub-nanomolar range, a very good value. Of course, affinity is not everything. Therefore, we performed a full lead discovery project to demonstrate the capabilities of the Pioneer platform. For this example, we decided for TIGIT, a checkpoint inhibitor with a potential role in cancer immunotherapy, with over 30 clinical trials ongoing. We selected 395 ELISA-positive hits, performed K-off rankings, sequenced 128 clones, of which 112 were unique, so a very high diversity. Of these, we expressed and purified 63 in Fab format and then further characterized these. We compared the Pioneer antibodies to 3 benchmark antibodies from the clinic: vibostolimab from MSD in phase 3, tiragolumab from Roche in phase 3, and domvanalimab from Arcus in phase 2. Finally, we also demonstrated a very good developability of our candidates. Let's start with affinities. Again, we had a good number of antibodies in the sub-nanomolar range, with several candidates in a similar range as vibostolimab. We even found an antibody with an affinity of about 1 picomolar. Next, we tested for their ability to inhibit binding of TIGIT to its ligand CD155 in an in vitro assay. The first bar with 100% inhibition is vibostolimab in Fab format, and you can see that there are several candidates with similar potential. Interesting candidates were then retested in a full titration. Vibostolimab is shown in black, and it's clear that there are several candidates with similar inhibition properties. With the affinity and inhibition data, we could select 11 candidates for conversion into the IgG format. These were cloned into a mammalian expression vector and produced by transient mammalian expression. All candidates gave very good expression yields of an average over 200 milligrams per liter, which not only gives us sufficient material for further testing, but also correlates with good antibody properties. We tested the IgGs for functionality in a cell assay. If TIGIT is blocked, the cells give a luminescent signal. We started with the 3 benchmark IgGs, of which tiragolumab and vibostolimab performed similarly and better than domvanalimab. We continued to compare our antibodies to tiragolumab, which is shown here in red. As you can see, we found several antibodies which perform similarly to this phase 3 antibody. Cell interaction propensity was determined by SI-BLI. This assay is a simple way with small amounts of material required to determine whether the antibody might give problems later when concentrated to very high concentrations, such as 100 milligrams per ml. We looked at 12 therapeutic antibodies in addition to the 3 benchmarks, and all our Pioneer antibodies gave low values in a similar range as these therapeutic antibodies. Let's look at all parameters and compare these to the 3 benchmark antibodies. There's a good number of Pioneer antibodies with similar or even better affinities than vibostolimab. The first melting point is for the CH2 region, and since all antibodies have the same CH2 sequence, there's no difference in these values. More interesting is the second TM value for the Fab domain. All antibodies have very good values of 73 degrees Celsius and higher. If we look at the monodispersity by SAC after a single-step protein A purification, all but 2 had values over 90%. Most were above 95%. Cell interaction propensity was very good and similar as for the benchmarks. One antibody showed a strong hydrophobicity by Higg. All others had low hydrophobicities comparable to the benchmarks. 2 antibodies showed an increased polyreactivity or polyspecificity, which was measured by binding to heparin, DNA, LPS, and CHO lysate. All candidates passed well the 4-week stability study at 37 degrees. Several candidates have EC50 values in the same range as the benchmark antibodies, as shown on the previous slide. And finally, the germline homology of these antibodies is in a very good range, above 88%, and even exceeding those of vibostolimab. In conclusion, with the Pioneer library, SpyDisplay, and the Trailblazer technology, we could generate and characterize lead candidates in less than 5 months, which show similar performance as therapeutic antibodies in the late clinical stage. And this directly out of the library without further optimization, whereas almost all antibodies in the clinic went through further optimization rounds. In summary, the Pioneer Antibody Discovery Platform comprises one of the largest functional phage display libraries ever made, with a minimized number of sequence motifs of concern and a curated CDR design. Our proprietary selection technology, SpyDisplay, which is very versatile and powerful for the selection of high-affinity antibodies, and the Trailblazer modular antibody assembly technology for rapid screening and testing in various formats. The outcome is a very good hit rate and sequence diversity, with over 20% of the antibodies with affinities in the sub-nanomolar range, and very good developability parameters. Candidates with similar properties as advanced stage clinical trial candidates can be selected directly out of the library. Thank you for your attention. Please submit your questions for the Q&A session at the end of the presentation.
Thank you, Francisco, for introducing us to the Pioneer Antibody Discovery Platform. I'm now delighted to introduce Dr. Paul Royle, who's going to be explaining more about the antibody discovery process. Dr. Paul Royle is a technical sales manager for Bio-Rad's custom antibody service and has been with the antibody division for over 9 years. Prior to this, he worked in an immunology diagnostics company for almost 7 years. He holds a degree and PhD from the University of Nottingham, UK, and has postdoctoral research experience from the University of Warwick in the UK. Now I'd like to pass you over to Paul. Thank you.
Thank you very much for the introduction, Kim. So yeah, my name is Paul Royle, and I am one of the technical sales specialists for Bio-Rad's custom antibody services. So essentially, the role of the technical sales specialist is like we're field-based application specialists, essentially, who specialize in the custom antibody service. So our role is to introduce Bio-Rad's custom antibody generation services through technical presentations, then to engage with customers in project-specific technical discussions to really understand the technical scope of the project and the requirements that the customer may have. We then basically capture those requirements in documents, we call them quotes, but essentially they're contracts between Bio-Rad and the customer. And these lay out both the technical and also the commercial details of any particular project. So there's a team of us, we cover the whole world between us. And although the Pioneer service is a new service, we have been offering HuCal generation services or tool antibody generation services from phage display platforms out of our lab near Munich for nearly 20 years now. So there's a lot of experience within the team about how phage display libraries can be used and the various strategies that can be employed to generate antibodies to meet particular requirements. So what I would like to do in these next few slides really is to give you a bit of a flavor of the types of discussions that people like myself will have with you as customers, and the sort of considerations that we take into account when we're putting together all of this information to populate one of these contracts. So I'd like to go through this in a little bit more detail. So I would like to start by really explaining the positioning of this service and where we fit into the whole workflow of drug discovery. So what we're essentially offering is custom antibody generation services. Our expertise is in building phage display libraries, generating antibodies from phage display libraries, characterizing those antibodies and further engineering those antibodies. And really, we're sticking with those areas of core competence. We are not really a drug development company. We don't do animal work. This is all in vitro antibody generation with characterization in vitro and using cell-based assays, etc. So there is the antibody discovery service itself, generating antibodies from the library. And in the next few slides, I'll go into more details about how, the considerations around that, because that is really the core of the service. There's a characterization of those antibodies, what sort of tests we're going to employ. When I talk about antibody re-engineering, there's obviously format conversion and the fairly basic types of re-engineering antibodies, but we also offer things like antibody affinity maturation services and things, and we have a lot of experience in that area. And finally, we appreciate that these will be drug candidates, and we have a long and proud history of generating tool antibodies to support clinical studies. So we're probably market leaders in anti-idiotype generation. So if we do generate leads or candidates, we can also supply the accessory reagents to help customers when they come to doing things like taking pharmacokinetic measurements of those drug antibodies in human serum, or if they need calibrators and controls for anti-drug antibody responses. So in terms of the commercial positioning of this service, essentially we limit our involvement to some extent. So what we offer really is a fee for service. All the IP around the new Pioneer library is Bio-Rad's own IP, so there are no follow-through licenses to other third parties. Because it's our own IP, customers can own the program IP, so they can own the DNA sequences of the antibodies we discover. They can own all of the downstream IP, and that, assuming that they've paid the relevant milestones, they'll be able to use that IP with no restrictions from Bio-Rad on freedom to operate. From the commercial side, we understand that the process of drug development is very risky, and essentially we model the financial parts of any quote or any agreement to reflect that risk so that we share the risk with the client. So we only ask for a modest upfront payment, and then we start to realize the value we've contributed to those antibodies as those antibodies are increased in value to the customer. And this is usually through developmental milestone payments when they hit various performance milestones, such as first in human or launch. What we don't ask for is royalties. So when you receive a quote from us for a drug antibody development program, essentially you can see what the maximum expenditure would be in a best case scenario where the antibodies we've generated for a customer end up as approved drugs on the market. So what I'd like to cover in this slide really is to give you an outline of the scope of the conversation we tend to have when we're discussing the technical aspects of a project with a customer, and also to give you an idea of the number of antibodies that we're talking about at each stage in the part of the workflow that really this conversation relates to. So with any project, one of the first things we need to establish is what materials will need to be used in the project. So most of the target antigens customers come to us with are proteins, whether they're soluble proteins or membrane-bound proteins, but really understanding what those proteins are, whether they're commercially available, whether they are already manufactured and made by the customer and are available from the customer, or whether they're something we maybe need to generate ourselves, or whether we need to make peptides to particular regions of those proteins. But it's not just the proteins themselves. In many cases, customers will also want to understand whether whether the antibodies cross-react to species homologs of those proteins, such as versions of the proteins that are present in relevant tox species. There's also cells, if cell binding is important for that particular antigen. Matrix is something we use a lot. So some antibodies are negatively affected by certain matrices, and the nature of phage display is that you can spike those matrices in, so that any antibodies that are negatively affected by the matrix are not selected during the antibody generation process. And then if we're needing to think about additional assays that we're going to start to look into, we need to start to consider, what those assays may comprise of. Are they receptor-ligand interactions, etc.? So do we need the ligands for the receptor proteins? Are there protocols for these assays? Are these assays available as commercial kits? There's a whole number of things to cover here, but really this helps us lay the foundation for how we're going to screen our library. And in the top right-hand corner, you probably heard this in Paco's talk earlier, is the sort of absolute size of the library in the unfiltered state, which is 225 billion antibodies unique by next-gen sequencing. The next consideration really comes down to the success criteria. So by this, we mean the success criteria in terms of what characteristics the antibodies we generate have, or what the requirements are for those antibodies from the customer. And the reason this is important and an area where a lot of consideration should be given is because the nature of antibody phage display and biopanning is it's a fully in vitro process, or it certainly is at Bio-Rad. And there are lots of ways in which that process can be manipulated to achieve particular desired outcomes. And understanding the success criteria and I suppose the hierarchy of importance of different things that comprise those success criteria can really influence the way in which the library is screened and the processes that are employed. For example, what cross-reactivity is undesirable versus what cross-reactivity is desirable. To give you an example, it may be that cross-reactivity to different protein members of the same family, which are very highly conserved, is undesirable, whereas cross-reactivity to a species homologue in a relevant tox species is desirable, because these are all things we can build into the way in which the library is screened to generate antibodies so that we only enrich for antibodies that have those characteristics. There are various ways in which phage can be eluted from immobilized antigens as well. It doesn't just have to be by protease treatment. So if we know, for example, a particular target, is small and in solution, we can try and elute our phage from the immobilized target by competition with that free form of the protein. We can use specific physical conditions as well, such as pH, to generate antibodies with particular characteristics in that area. And we don't just have to work with proteins, we can work with peptides, we can work with whole cells as well. We can deplete our library for antibodies to the host cell, for example, a CHO cell, by doing a pre-incubation block of our library with non-transfected CHO cells before then screening on CHO cells that are overexpressing a particular cell surface target, for example. So really, we try and understand what the customer wants to achieve, and we're not prescriptive in terms of the strategy that we put into the contract, but we do define what those success criteria should be so that the lab can work to achieve those criteria and try multiple different strategies if they deem that appropriate. So really, it helps us to understand what are the exclusion criteria, what are absolute requirements, and what is desirable. And just to maybe elaborate this point, consider a case where you're a customer, you want us to generate an inhibitory antibody to an enzyme or a receptor or something, and there's another antibody out there that is inhibitory, and we're able to obtain that antibody or make it and use that as a control in some of our experiments. Now, if that inhibitory antibody, if you're free to use the IP around that, then it may be that you want inhibitory antibodies and you don't care whether they share the same epitope as the existing inhibitory antibody or not. In that scenario, we would generate inhibitory antibodies and then we do epitope binning afterwards to tell you whether they were competitive or not. And in an ideal scenario, you get both competitive and non-competitive inhibitory antibodies. However, if there was IP around the epitope recognized by that particular drug antibody in that particular application, and you wanted to generate an inhibitory antibody to a different epitope, we could use that existing antibody to mask the epitope that it does recognize and then we could screen the library for inhibitory antibodies against that masked antigen, so that any inhibitory antibodies we generate would be to a different epitope. So understanding this can really affect the way in which the library is screened. And all of this will be done to really reduce that number of antibodies down from the library of 225 billion to sort of thousands of individually expressed proteins. So these will be expressed as antibodies at the protein level, and then these can be screened and further characterized, and the selection of the best of those binders made in the next step. Another advantage of a fully in vitro process is that you have a large amount of time in which to employ different screens throughout the antibody generation process. So the nature of our workflow is it can literally be frozen down at various stages and tests done on expression plates to really characterize antibodies at the protein level without having even sequenced them at that point. The nature of our workflow is that from the tens of thousands of antibodies we get out of the enrichment process, we pick individual colonies into multiples of 4x3 Orwell plates. Those antibodies are expressed as proteins. They're characterized, and then antibodies that look like they have promising characteristics are then sequenced and expressed as pure proteins and further characterized. So there's a lot of screening we can put in at various points in the process, and it doesn't just have to be binding assays, i.e. does it recognize what it should, does it not recognize what it shouldn't, but we can also start to look at early functional assays. So, we can look at whether antibodies appear to be inhibitory. We can look at the off rates of those antibodies to try and identify those with the best affinities. We can do applications like Western blotting and flow cytometry, and using a technology we have called SpyTag, we can even look at these in both the monovalent and the bivalent format. So the most stringent combination in which we use this is, does the monovalent version of the antibody give good binding to the target protein, and then does the bivalent higher avidity format still give a very clean reading with no undesirable cross-reactivity creeping up as you go from a relatively low avidity monovalent format to a higher avidity bivalent format. So really it's this process that really helps us whittle down all of those antibodies to understand which ones are the best to carry forward to the next stage. So from the hundreds of E. coli expressed antibodies, we really need to make a decision about which ones to carry through to fully mammalian expressed human IgGs that will essentially comprise the deliverable to the customer. And what we try to build in here is quite a lot of characterization, both at the E. coli level and then at the mammalian level. So we're both confident that we're really picking the best clones, and then also that we give a lot of data to the customers, so they're confident in taking these forward into their further studies. And the number we convert to mammalian expressed will really depend on the customer's ability to sort of really characterize in more detail these numbers of clones. So we're usually in the tens of antibodies by this stage. But we can do all sorts of screening at Bio-Rad, all sorts of characterization, assuming we don't need to do further re-engineering. And this could be, looking at the sequences for developability concerns and homology to germline, and then a lot of developability tests such as melting point determination, self-interaction, monodispersity, and various screening assays from polyreactivity, polyspecificity assays for developability, and also just confirming the final characteristics of those antibodies, all the specificity, the affinities of them, the epitope binning. There's various things we can do. So this will really sort of complete the project. Then we will transfer a number of antibodies with the desirable characteristics to the customer, and that will be essentially the end of the project unless they want further services from us. So really that brings me to the end of my presentation. I hope that's given you a bit of a flavor of what a service from us would look like, and I'd be happy to take any questions. Thank you for your attention.
Thank you, Paul, for explaining how the Pioneer process works. And thank you to all of you for joining us today to learn more about the Pioneer Antibody Discovery Platform. We've now reached the Q&A portion of our webinar. You can submit any questions that you'd like us to answer by using the Q&A widget. Any questions that we don't have time to answer, we'll follow up on after the webinar via email. Thank you. Thank you everyone for your questions. Our first question is, in Pioneer, is only HCDR3 randomized or are other CDRs also randomized? Francisco,
Yeah,
can you answer that?
In the Pioneer library, all 6 CDRs are diversified with a germline-specific design.
Next question is, where do the Fab and phage assemble in E. coli in preparation of the phage library?
So the Fab with the spy tag, the heavy chain, the light chain are transported into the periplasm where they fold, and the P3 with the spy catcher is also transported into the periplasm, and there the protein ligation will take place, and the Fab is assembled.
Thank you, Francisco. And does the Fab in Pioneer have a disulfide bridge between the light chain and the heavy chain?
No, we do not have a disulfide bridge between the 2 chains.
Okay, and someone would like to know how you do the affinity maturation with the Pioneer clone and if it's the same as HuCal, like full maturation or wrap mat with conversion of cassettes for each of the CDRs.
So for the affinity maturation, we actually redid the complete protocol. So we use now SpyDisplay as well for the affinity maturation, and we have a new method to generate the diversity, the maturation library by conchyl mutagenesis. So we have shown for the maturation of Pioneer binders that we can reach affinities below 1 picomolar or in the low single-digit picomolar range. So the affinity maturation works very well, and the new protocol delivers very high affinity binders, which basically shows again that the SpyDisplay selection technology is very powerful for the selection of high affinity antibodies, and that our library generation protocol works also very nicely. We can diversify the heavy chain CDR2 CDR, the light chain CDR3, or both CDRs in parallel together.
Thank you. Another question is, Trailblazer seems like good technology to screen antibody drug candidates. Can Bio-Rad do custom payload conjugation onto SpyCatcher?
Yes, that's something we certainly can discuss, and we can discuss the requirements and then perform conjugations for our customers.
Right. And there's another technical question about how do you elute the bound phage from the antigen, and is this different from SpyDisplay?
Yes, it is different. In SpyDisplay, we have a disulfide bond, which we can cleave with a reducing agent like DTT. In Pioneer, in SpyDisplay, we have a covalent isopeptide bond, so it cannot be cleaved that easily by adding a reducing agent. It's a stable covalent bond. So here we introduced the protease cleavage site so that we can use a protease to cleave the specifically binding antibodies from the phage and elute the phage like this.
Thank you. For all the next questions for you, who owns the antibody sequences of the lead candidates generated with the Pioneer platform?
So this would be something the customer can own in a project. So all the IP was generated by Bio-Rad. So there's no follow-through licenses to any third parties. So yeah, provided a customer is, is meeting the terms of the agreement, making the correct financial payments, then they can own the physical antibody, they can own the sequence of the antibody, they can patent that antibody, and there's no restrictions from Bio-Rad in how they can use the antibody.
Right. And this is a question, Paul, about whether Bio-Rad's biotherapeutics antibody discovery service, Pioneer, is available straight away for projects.
It's available. I mean, currently it's launched. We can start projects. In terms of how quickly we could start from a first discussion, it would be, it really depends. I mean, what we tend to find is the technical discussion and the contract side can take a while to negotiate, so that gives us plenty of time to schedule the actual start of the work. But yeah, I mean, if customers have an urgent requirement, we'd be talking weeks rather than months probably before we could agree a start date if there is something very urgent. But as I say, the service is available now. So yeah, if customers do have a project they're interested in, please come and talk to us.
Great, thank you. Francisco, someone wants to know what are the main technical advantages of Bio-Rad's Pioneer platform over other phage display antibody libraries?
First of all, it's the size, but it's not just the huge number. It's especially that the sequences are optimized for a well-balanced expression in E. coli and selected for suitability for phage display. And that's basically what makes the Pioneer library one of the largest functional phage display antibody libraries. But that's not all. The sequences were designed to result in antibodies with good developability properties, as we have demonstrated for the TIGIT antibodies. This reduces the timelines for the discovery of lead candidates and reduces the risk for costly optimization rounds.
Thank you. Someone wants to know who can be the potential customers for Pioneer.
I can take that one if you want. So the potential customers for Pioneer, we envisage they'll mostly be companies developing drugs just because of the price of the service. So we imagine that, but there's no restriction from us on only working with certain customer classes. But what I would say is if you need just research antibodies or antibodies for in vitro diagnostic use, again, generated by SpyDisplay, we do have a QCal custom service. So this is a tool antibody or a research antibody generation service, which is it's basically cheaper because a lot of the characterization of the antibodies that you need for a drug is not necessary and not performed. So we have like one library which we use mostly for tool antibody generation, and then the Pioneer we envisage what we primarily use for drug antibody generation. But if the customer is willing to pay the price and wants Pioneer antibodies for non-therapeutic applications, we can certainly look at that. I hope that answers the question.
Thank you. And someone wants to know if they're interested in discussing a potential project, would this be done under a confidentiality agreement, and who they should they reach out to find out more?
Yes, I mean, at some stage it will almost inevitably end up with discussions under CDA, but if customers want an initial conversation where they don't necessarily want to disclose the protein that they're targeting or that they would need a drug antibody to, we can, they can tell us it's a soluble protein or it's a membrane-bound target, and we can have conversations in general about it. But I think to have a meaningful conversation and to really get into the technical details, it helps the customer to have a CDA in place. It's not necessarily a requirement from us, but most customers don't want to disclose the biomarker they're targeting in a drug project without having that covered by a non-disclosure or a confidential disclosure agreement.
Thank you, Paul.
But I should say also that we can arrange a two-way confidential disclosure agreement very quickly. we have standard legal documents for that.
Right, thanks. And we have one final question, which is for Francisco, which is, did you use next generational sequencing to control for the Pioneer library quality?
Of course, we used NGS to verify and confirm the high quality of the library. We have a very low error rate, such as stop codons and frame shifts, etc., of less than 10% of the sequences in the Pioneer library. And the actual library sequences are very close to the intended design, basically confirming that we have a high quality synthesis and cloning strategy. And basically we also confirmed that we have a very high diversity.
Right. Thank you very much, Francisco, and thank you, Paul, for your presentations today. And thank you, everyone, for joining us. The presentation will shortly be available to rewatch on demand if you wish. Thank you, everybody.